Today in the class we are doing these hydrolysis tests for our unknown bacteria. So one thing that we want to remember here is that we are basically testing for the exoenzymes. Okay, so we want to see whether our unknown bacteria is producing these three different enzymes or not, and they are called exoenzymes. Okay, so the first test that we are doing is called a casein hydrolysis test, which we use by using the skim milk plates. The second test is the starch hydrolysis test, which we will use by using the starch plates. Okay. The third test is the lipid hydrolysis test, and the lipid hydrolysis test we are going to do by using these methylene blue plates. Okay, these methylene blue plates, they look slightly blue in color because we have methylene blue dye added to it as a indicator, so that it can show our results, clearing results a little bit more pronounced. Okay, so this is my skim milk plate, and the skim milk plate, it will kind of look little whitish because it has a small percentage of skim milk added to it. This is the starch plate. This also has some percentage of starch added to it. So what we are going to do is we are going to inoculate our bacteria right in the middle in a straight line, a little thickened line. That's how we are going to inoculate. Instead of doing a normal zigzag that we normally do, we are just going to inoculate our plates right in the middle without touching the top or the bottom of the plate. Okay, so the reason of doing that is simply that once we get our results after incubation, we are going to check for the production of these three enzymes. In case of skim milk, we are going to check whether our unknown has produced the enzyme called caseinase, because milk normally is supposed to have a protein called caseinase. I mean to say casein. So if our bacteria produced caseinase, then it is going to break down or hydrolyze the protein casein. And once it does that, you can see it as a clearing zone around your growth, around your bacterial growth. So this will appear as a clearing zone. Now for the starch test, it's slightly different because here, of course, we are going to see the ability of our unknown bacteria to produce amylase. Okay, if it produces amylase, then amylase enzyme breaks down the starch. Okay, so in this we want to test after incubation with iodine, because iodine is the detection reagent for the starch. Okay, if here it is slightly different. If after adding iodine, our plate still appears blue-black in color, that would be a negative test. Okay, that means starch is still present inside our plates. The bacteria could not produce the enzyme amylase, and that's why it could not hydrolyze the starch, and starch is still intact inside the plates. Okay, the negative result will look like this, just your bacteria growing on the plate without any other color. But if there is no change in these plates, that would be your positive test. But if your media turns blue-black, that would be a negative test. Okay, because starch is still there and it is going to stain blue-black with the iodine. Okay, Now let's talk about the lipid hydrolysis. In case of lipid hydrolysis, you have methylene blue plates, and we are going to test the ability of our bacteria to produce the enzyme lipase. Okay, so if this enzyme lipase is produced, then in these methylene blue plates you have a small percentage of lipid also added. So if the lipase enzyme is produced by your unknown, it's going to break down the lipid, and you can see that again as a clearing zone around your bacterial growth. So this clearing zone will indicate a positive test. We are going to know these results on Thursday after incubation. We are going to incubate all these plates at 35 degrees Celsius so that we can read them on Thursday. So apart from these hydrolysis tests, we are doing one more additional test, that is we are going to check for the enzyme urease. Okay, if our unknown has produced urease enzyme or not. In this case, all we are doing today is inoculating the urea broth, which looks like this. So this urea broth, which is slightly pink in color, okay, we are going to just simply inoculate with our inoculating loop. We are going to inoculate our unknown just using the aseptic technique. We are just gonna put the inoculating loop inside with our bacteria. Okay, so if your bacteria turns out to be urease positive, it will change the color of the phenolphthalein that is present inside it into a hot pink color. But that result you can see only after incubation. So right now we can just show you the negative or the control tube. Okay.